Welcome to the Good Medicine Podcast. We're very excited today with our episode to have Chris Gibson here with us. I guess I should call you Dr. Chris Gibson, but
Chris is great.
Chris is the co-founder of Recursion Pharmaceuticals, and Chris holds a PhD in bioengineering from the University of Utah. And Chris and I met a long time ago when you were a student, and I knew Chris was destined for big things when I first met him. And Recursion Pharmaceuticals has really become a real leader in the use of artificial intelligence to enable drug discovery. And so we're so excited to have you here today and to have a discussion about, there's a lot of hype out there about what AI is and what it does. And we hope to dig in and talk a lot about, what is AI and how do you use it and how does it enable drug discovery. But before we jump into that, I think it's always great to hear about you personally and your background. I find it fascinating of how everyone ends up where they are. So as a child, did you dream about being a scientist, or was your mom and dad? So there's all kinds of stories. Yeah. So I'd love to hear. What is it in your life that led you to want to be involved in science, and specifically what got you excited about being involved on the therapeutic side of things? Yeah,
well, first, thanks for having me on. It's really exciting to be here, and I'm excited for everything that's happening in Utah. So this is a lot of fun. I was one of those kids who took everything apart when I was a kid and then tried to put it back together. I always loved to understand how things work, and I was pretty into science at a young age. But I thought I was going to be an astronaut.
Nice.
And then in, I think, 6th or 7th grade, I passed the height limit.
Oh, okay.
So at least I heard at the time it was like 6 feet or 6'1" or something. Yeah. I passed that. And so I had this existential crisis in middle school. And had to figure out what to do with my life because astronaut was off the table. I always was into science. And when I went to... When I was thinking about what to do, I talked to my dad a lot. He had built a company starting 2 weeks before I was born, and he was at the interface of electrical engineering and business.
Okay.
And so he built a company that was based on some of the first parallel processors that came out of Intel. And so I got to see that happen as I grew up, and I was like, I kind of want to do something like that. But I thought that the next big wave was going to be in bioengineering instead of electrical engineering. And so that's what I went into in school, and long serendipitous path of how I ended up in Utah, but I ended up in the lab of a guy named Dean Lee, who you knew.
Yep.
And it was a project in Dean's lab that led me to join you up at the Huntsman for some work together in drug discovery. And one thing led to another, and Recursion came out of that. So a long path, a lot of serendipity, but I think in some ways always destined to be in the interface of different fields. I'm not very good at any one thing.
Yeah,
but I can do a few things reasonably well, so it works out.
Well, I can completely understand that, and I feel the same way. So that's exciting. So you're a graduate student. You're working in a really great lab with a great mentor. What was the beginnings of the thought? So you kind of had this background where you were exposed to your dad being an entrepreneur. Yeah. As being a trained scientist, we really don't get trained to think about starting a company as kind of a career path. I know I was the world's most reluctant entrepreneur. I was a faculty member and had a grant and all this stuff, and that's the world I knew. So what was it that kind of flipped the switch for you to say, hey, I could actually take this and turn this into something that could actually be a company that I could build around?
Yeah, I had actually in grad school gotten a little bit pulled away from the entrepreneurial side. Yeah. And I was doing an MD PhD, and I thought I was actually going to be a CT surgeon.
Okay.
In Dean's lab, one of the things we got to do was hang out with other people in the lab, and there were a few cardiothoracic surgeons there. And so I would go with the pager at 2 a.m. when they did a heart transplant, and I would get to basically just watch and sometimes help collect tissue from the hearts that came out of patients. Fascinating. And I thought entrepreneurism is really cool, but this is my future. And then we were working on a project in Dean's lab on a rare genetic disease called cerebral cavernous malformation. And it was one of many projects that I got to be a part of as an engineer in his lab. And ultimately the traditional approach that we took failed. And so we decided to try a different approach and use some basic machine learning classifiers to look at images of human cells. And so you've got images of human tissue here.
Yeah.
So we used a machine learning classifier and made some progress in a place where we hadn't made progress before. And that just felt like it was something we had to pursue more. And so in speaking with Dean, he arranged for me to go to Stanford for the summer and do sort of a mini entrepreneurial project there. And that just snowballed into what would become Recursion. And it felt like it was one of those things I was going to regret if I didn't try. It felt like the most likely outcome was failure.
Yeah.
And so worst case is that I was going to lose a year before I went back to med school, but I was going to gain a lot of experience. And so we took the shot. And I think leave of absence from medical school in 2013. And like by 2016 or 2017, I wasn't back, and they made me formally drop out of the program. So I'm a med school dropout too, which plays very well in Silicon Valley.
I bet it does.
When you're raising money.
Yeah, that's right. Well, that's wonderful. So tell me about, I've always been involved in starting companies and putting that pitch together of this is what's unique about us and our approach. So as kind of a pioneer, really a pioneer in the area of adapting machine learning and artificial intelligence to drug discovery, what was the original pitch that you came up with that got people interested, and how did that morph over time, and how did it resonate with the early investors that came in to help you get going?
Yeah, it was interesting because there were not a lot of investors at the interface of biology and technology at the time. So that we were kind of like looking for biology investors or technology investors who could see across fields. Mm-hmm. What was super interesting is that in 2012, there was this thing called ImageNet that was released. Google did this, really the first big sort of machine learning project on images, and it created a renaissance in computer vision. So can you essentially take an image and use a computer to extract really meaningful, rich representations? And so that was happening in 2012. Recursion started in 2013. Mm-hmm. And we had this evidence that maybe images in biology were important. And so the pitch was that in biology, structure suits function. Mm-hmm. And if we can generate a massive library of images of biology, and we can get really good at doing computer vision, we could decode biology. And the business pitch was that we were going to repurpose medicines. So there's all these thousands of medicines that people have tried to take to the clinic or to the market that have failed for a variety of reasons. And we're going to find what they should have been designed for.
Yep.
We're not doing that much anymore. Yeah. So pivot was that it turned out most of these medicines were shelved for pretty good reasons.
Yeah.
We found a few interesting indications, and we have clinical readouts coming for some of those in the coming quarters. But we found less opportunity at the interface of shelved assets and rare genetic diseases, which is where we were looking for a wide variety of reasons. And so the way we started morphing in those early days was we started doing our own chemistry too. Uh-huh. We found interesting targets or interesting pathways, and we figured we had to make the right chemical at that time. And so that's been a journey. It keeps the scope of what we have to build keeps getting bigger.
Yeah.
And so, we never thought we'd do our own clinical trials. Now we're doing a whole bunch of our own clinical trials. We never thought we'd have our own supercomputer. Now we have our own supercomputer. So it's become just a bigger and bigger endeavor. But the core idea that you can use computers to extract meaningful representations of biology from data-rich, scaled, data assets, that's held true. And it's been a lot of work, but we're here 11 and a half years later, still doing that same basic thing.
That's incredible. Yeah, and I think it's it's fascinating because it is one of the central tenets of biology is structure matches function, and that's not just at the molecular level, that's at the cellular level, which is really what you're...
back when physicians used to really take time with their patients. Yeah. I mean, I read a lot of William Osler and some of the incredible learnings that physicians back in the day could take just from looking at little things.
Exactly.
Right? The little dots on your lips or your tongue or the curvature of your nail beds. Yep. These sorts of things tell us so much about biology. Yep. And I guess it's not that surprising to imagine that the same thing could hold at the level of cells.
Yeah.
And that we found it to be true.
Yeah.
I mean, we've now knocked out every gene in the genome and many different cell types. We're not only doing that work for ourselves, but doing that work with companies like Roche Genentech and Sanofi and others. And but it also turns out biology is super, super complex. Yeah. And so like everything, I think we're making incredible progress, but it's a very, very dense, challenging, complicated map that we're trying to build.
Yeah, and I think that's the biggest challenge because most scientists want to take the simplest thing and so we find a single gene or a single protein that we think is changed, and we put that in a test tube and study that in isolation. But the reality is, biology is never one protein acting by itself. Very rarely,
I think that's a great way to do it too. In algebra, you try to minimize the number of variables, and that's how you solve the equation, right? And so that is a very, very viable path, and it's how much of medicine, there's sort of been two paths, like that path, or kind of serendipity and luck. Like you drop the wrong test tube in the wrong vial.
Oh, look what happened.
Something happens, right?
Yeah.
That's kind of how most medicines came to be.
Yeah.
And it felt like technology was getting to a point where, and the tools in biology were getting to a point, things like CRISPR were coming out, and then also the computational side of things. I mean, if you were to do what we're doing today at Recursion 15 years ago, you'd have to spend $1 billion just on compute and data storage. So it just wasn't feasible.
Yeah.
And so all these things were starting to come to fruition at the same time. And the question a lot of people ask, I think now many people think this technology-driven approach will be an important part of the way many medicines are discovered in the future, not all. And the question is were we on one wave too early? Were we on the right wave?
Yeah.
We're probably not too late.
Yeah.
We're still one of the earliest ones in the space. So it's a question of too early or right at the right time. And now there's hundreds of companies and most large pharma that are putting real resources into this idea of leveraging technology to look at large, high-dimensional data to take a more complicated view of biology. And in many ways, you still need all those tools of sort of this reductionist approach. And putting those two things together, I think there's going to be, hopefully, an explosion of more medicines or better medicines in the coming decades. That's what all of us want.
I think that's exactly right. And I think that the idea that we can add on to kind of the traditional way of doing things with something that's, because the hard thing about biology is it's so complex, you can get lost in that complexity really easily. But the way that Recursion's taken that approach to say, we're going to start with that if when we have a disease cell or a diseased tissue, it looks different, right? And that look is actually the structure of that cell actually tells you something about what's going on in the function. So I love that idea that helps reduce the complexity of biology. You say, this is one thing we can track and we can measure and we can say, this is the result of a bunch of complex biology, but it's something we can wrap our heads around, right? Because a lot of times, you've looked at it, and I have, transcriptomics and RNA-seq data. I mean, it just blows your mind. I don't know how to make sense of this stuff. But I love the approach that you've taken where it's something that at least I can conceptualize to say, all this complex biology results in this, and I can measure this. And correct me if I'm wrong, but I think what your approach to drug discovery is really, can I correct that to make that dysfunction look normal again, right? So if I can take a cell that is taking on this abnormal function and abnormal structure, can I restore it to look like it's normal again? And therefore, I should have fixed all that biology that was messed up.
Yeah, that's exactly how we started and what we still do. What we've added to that is not just these images, but also transcriptomics and data from patients, all this rich high-dimensional data, which machine learning and AI are actually pretty good if you set it up right at looking across really large data sets and finding these patterns. What it turns out happens is if I take an image of one cell and one disease state, I can describe how that cell looks different. It turns out computers are way better at that than we are. They can see way more subtlety than we can as humans. But I don't really know what's happening. I know that the cell looks different. Maybe I broke a gene, it looks different, and now I add drugs and maybe one of them makes it look healthy. That seems good. But as you start doing this across millions of different experimental conditions, millions of different small molecules, millions of different genetic perturbations, you actually kind of build this lookup table. And this is the part that gets most exciting to me when I talk about mapping biology, is now instead of saying on a disease by disease basis, I can ask if I break a gene and the cell looks different in some way, across all other ways I could break genes, do any cells look really similar? And that can actually start to tell you something about the proximity of these really complex pathways, like does gene A interact with gene B? And you can start taking this a step further and asking, can I predict essentially what all of the pathways in biology look like in various cell states? And can I then correspondingly place small molecules or large molecules in all of those different areas of biology? And that's kind of why this experiment of recursion has grown so much with supercomputers and massive automated labs, is because we saw the opportunity to maybe find some really exciting medicines if we were doing this rescue the disease state. But moving into this mapping and navigation approach, this is like a boil the ocean project.
Yeah, for sure.
I mean, with Roche Genentech, we had to make like over 1 trillion neural iPSC cells in order to build this neural map. We think we became the world's largest producer of neural iPSCs just to make that one map.
Yeah.
Right? Like we called a supplier and they said it was going to be between $750 million and $1 billion to buy the cells from them. And our budget was like $7 million.
Yeah. So how do we do
So
that?
we're like, okay, I guess we'll do it ourselves.
We'll grow it ourselves, yeah.
And so like it's become this really big project. But the more you build these data sets, the more connections, not just we can see, because it's too complicated, the computer can see. And then it gives us interesting associations that we can then take into these more traditional approaches and try and take medicines forward.
Incredible. BioHive is a nonprofit building the future of healthcare innovation right here in Utah. As one of the nation's fastest growing life science industries, they bring together scientists, entrepreneurs, students, and companies to create breakthroughs that save lives and strengthen our economy. Whether you're in a lab or at a startup, BioHive is here to build, brand, and bring together a thriving life science community, and there's a place for you. Learn more, get involved, and see their latest happenings at biohive.com. So I guess that sets up perfectly with the next question I have, which is, there's a lot out there, and you were one of the first, and so you've seen this whole field kind of grow of how do you adapt these machine learning. And by the way, tell me what the difference between machine learning and artificial intelligence, because I think I know, but you probably know better than I do. But how do we adapt these approaches into biology, and what is the potential? You probably saw in 60 Minutes last week, the CEO of DeepMind came out and said in 10 years, every disease will be cured. And you kind of hear that stuff, and you and I probably sit back and say, that's a little bit of a bold statement, but I love the vision that says, we're going to make a difference, and I believe it. I think that we're at the beginning of a revolution that's just going to get bigger and bigger. But help us see through what the real promise is, and what a little bit of the hype that's around, because obviously things get a little out of control sometimes. And I've heard it many times in my life that this is going to be the next thing, and it kind of doesn't ever live up to its potential. So
I mean, it's biology is pretty humbling. I've learned that. I've been guilty of some of these things. I mean, it's possible I was on a podcast roughly 10 years ago where I said we'd have 100 treatments in our pipeline within 10 years.
I kind of remember that.
I think we've got like, between us and our partners, 30 or 40.
Okay, well,
so we're making progress.
Sure,
but definitely less than I expected then. And so, look, I think when you see the power of these technology tools, even back 10 years ago when we saw the power, it makes it really hard to feel like there's not going to be an exponential shift in this field. And the question is where we are on that curve. And I think, as bold and ambitious as the comments that Demis made were on that 60 Minutes episode, I do think we are somewhere in the range of seeing things that are that crazy different than they are today in some number of decades, right? Whether it's 10 years, I thought it would be kind of closer to now, and it's a little slower than expected. Demis thinks we're 10 years away. I'm probably closer to Demis than I am to Derek Lowe, who wrote an interesting rebuttal to Demis's comments. Derek's perspective, and he wrote about what I said 10 years ago. And he's probably surprised to hear we're still around and kicking. But, I think his comment was that ML and AI are not capable of generating any new knowledge. And I think that is completely false. I think they are now very capable of generating new knowledge. We see it all the time internally. I think our colleagues at Roche and Genentech see the new knowledge that is coming out of our work, Sanofi as well. So I think that's demonstrably false. Where we are on this curve is discovering and developing a medicine isn't one step. It's not finding a target or then finding a molecule. You got to go through, ADME, talks, translation, clinical trials, et cetera. And I think that's what's slowing us down. I think if you were to ask to pick one step in drug discovery, can we achieve what Demis or I thought was true? I think we're on in 10 years. Absolutely. The question is, how do you stack that up with all of the 100 things that are required? As you know better than I, to actually make a medicine, right?
Yeah.
You've done that. I've not yet done that. And so you've got to deploy these technologies across hundreds of steps, and putting those all together and stitching them all together and making them play nice, and just the infrastructure required to build the data sets, like that's a lot of work. And I think that's why it's taken longer than I expected. And biology's hard, but I think we're closer to Demis's vision than we are to Derek's.
Yeah, I would agree with
Sorry,
that. I generally certainly agree with that. So I guess the question is, so developing all these tools and getting all the front end right, if the back end isn't ready for the push through, I mean that's where you run into these obstacles. So you said it earlier that you didn't think that you were going to be running clinical trials and doing all these things, but now you are. So I guess the question is, are you finding yourself thinking about, well, how do I take the approaches that I've taken on the discussion? Am I going to start making AI for clinical trials? And we're going to, is that something that's on the horizon for you guys?
We are doing that now.
Perfect. All right.
Yeah. We're getting frustrated. I mean, clinical trials are hard.
Yep.
There's not a ton of data, but there are some companies now who are starting to aggregate more data in the space. So a good example is we have a partnership with a company called Tempus, who's taken a lot of data from oncology patients. And any one of those data sets by itself is maybe not that useful. But when you look across tens or hundreds of thousands of patients, you can start to see interesting patterns. Like if I had a patient who had this particular form of breast cancer and they got this particular medicine, what was the outcome for that patient? As you start looking across tens of thousands of patients, we start to see really interesting patterns. And when you then pair them up with this kind of perturbative biology platform that we've built, and you put AI across all of those, we see the opportunity to do things like define the patient population better for your clinical trials. Yep. And then we also are starting to doing clinical trial simulations with machine learning and AI, and that's helping us sort of be more adaptive in the way we drive. The challenge, of course, is each clinical trial is tens of millions of dollars. And so unlike an experiment in the lab that might be 5 or 10 cents, and you can try millions of different versions, clinical trials,
you get one shot.
You get one shot. And so it's a high-risk place to innovate. But we hired this woman, Najat Khan from J&J, who was their chief digital data science officer. She had been, I think, visionary in applying these tools to not just clinical development, but actually commercialization at J&J. And she's a trained chemist. She knows the front end as well. And she's been helping us build out the tools in that space. We're very early.
Yeah.
And some of it is like not AI or machine learning at all. Some of it is like basic software engineering that's just automating how we email like sites back and forth. So we don't have to have 50 people on our clinical operations team.
Right.
But there's some low-hanging fruit.
Oh,
and very little has been built in the space. We were surprised to see. So this is the challenge. I mean, the infrastructure required to develop a medicine is massive. Yeah. And if you want to deploy technology at many of those steps, it just takes time. But it's, there's hundreds of companies now. You're building companies like this as well.
Yeah, for sure. So let me ask you, the next hard question is what about the regulatory side of things? And how has your interaction with the FDA been in terms of how do they feel about this idea of artificial intelligence, machine learning, and being involved across the process? Typically government organizations aren't necessarily the most nimble and the most adaptable to new things, but do you see promise there? And I guess the FDA right now, there's a lot of uncertainty of what it's actually going to look like, what it looks like now and what it's going to look like in 2 years. But in terms of your interactions, do you think that the regulatory is ready for something like this as well? And do you see signs of optimism that things are going to, because we obviously work in a very regulated environment. We can't just go do whatever we want. With clinical studies. What's your thoughts on that?
I think that the FDA has a pretty hard job, right? I mean, because if they go too slow, we get mad. If they go too fast, it's dangerous for patients, right? So they've got to walk a pretty tight rope.
And they're kind of damned if they do and damned if they don't, right? There's no win situation for them.
Exactly. And so we got to give them some credit for the situation they're in.
Yeah.
Our experience with them has been universally positive. I mean, they've asked us some questions or asked us to do some things that we didn't really want to do. Yeah. But by and large, it's been a lot better than I would say the folklore sort of told me it was going to be before we went to the FDA. Right. And now we've had, I'm sure 100 interactions with them. It's been good. What we've seen actually is the FDA is probably further ahead of many large pharma companies in this space. They put out a white paper, I guess, 2 years ago now, in May, that talked about the ways they were thinking about using AI in sort of the preclinical, clinical, and development side of things. It was, not super granular, pretty coarse, but it actually suggested they were thinking about it. And they just recently, a few weeks ago, talked about the ways they want to use AI for a variety of the kind of preclinical data that one generates. So I think that they're being pretty forward-looking, but they have this hard job of protecting patients, and it's an important job. And so I think it's probably like the onus is on us as builders in the space to go to them with smart risks, right? One of the things we like to do is go to them with, here's some predictions. And also, here's the wet lab experiment. And if we do that enough, I think over time, they're going to say, you know what, you guys have been pretty good with this, and other companies too. Maybe we're willing to just take the prediction in the future. But I think we've got to be, that's the one place where the transition might need to be slightly slower.
What if you could get rewarded just for being healthier? Meet Galvin, the company using the latest blockchain technology to reward you for taking care of yourself. They help with your mind, body, and soul like a personalized health assistant to track your progress and help you feel better. Galvin's G Plus app rewards you for exercise and assesses your mental health in under a minute using AI and over 2,500 voice biomarkers. If you're feeling off, G Plus gives you personalized power-ups, science-backed techniques to help you feel better fast. And here's the best part. When you prioritize your wellness, you earn crypto. Ready to level up your health? Use promo code GOODMED20 at checkout for 20% off. Download G Plus today at the Apple app or Google Play Store and start your wellness journey with Galvin.
No, I agree with that. And I think that I think a lot of people don't appreciate the fact that when you're thinking about revolutionizing this whole process, it is such a complex process of getting a drug approved, and there's parts of it that we probably do need to be more careful than others. I mean, at the front end, we can do crazy things and try the wild ideas.
In the lab, you can try anything.
Do whatever we want, right? Exactly. But you probably don't want to be doing that
unless it's like with gain of function, infectious disease mutations.
Let's not do that. That's right. Let's not create another pandemic. But I think that it's interesting to watch how all this is evolving. I'm hopeful. Obviously, I think being in science, I know you well enough. You're an optimistic person. I'm an optimistic person.
You have to be.
Yeah, you have to be. Like otherwise, we'd be just rocking back and forth in the corner because what I tell everybody, we put the in research, right? We have to do things over and over again, and there's so much failure that we really have to be optimistic people to think, hey, I learned from this, and next time, I'm going to get it right, and things are going to happen. So I look at what's happening right now, all the confusion and chaos, and I think a lot of it is probably things could be messaged a little better of what the end goal is here of all these changes. But the reality is, I think that the good thing about things changing is it gives us an opportunity to shape things in a new way. Because I really believe that we're in an inflection point, not just on the way we discover and deliver new therapies to people, but our approach to medicine, I think, is really changing as well. Because we used to think about, and we still do, obviously, that we treat sick people, and that's people can wrap their heads around that. I break my arm, I go put a cast on and go home. That's great. That's good medicine, right? Yeah. That's what we think good medicine when we think of good medicine. But I think we're coming to the point in the world right now with the demographics that are changing. We're getting older. With age comes chronic diseases, and we've not been really good at taking care of chronic diseases. They're not something you can wrap a Band-Aid on, send people home, and they get fixed, right? It's a challenge, and it's been one of the challenges is it's expensive to develop these drugs, and we have to recoup those costs. We have to provide returns to investors, and how far can we keep pushing this, and when does it break? And I have a feeling we're kind of to a breaking point in a lot of ways. And so a lot of these things that are happening right now, I think, are really being driven by, you look at Medicare's budget for the next 10 years, it's not good. Like there's not positive things happening. So I think it's forcing us to start thinking about, how do we keep people healthier longer, as opposed to, so good medicine might be changing a little bit of the way we think. It's always been that way, right? The best medicine is never get sick. Yep. I mean, that's the best thing that we possibly could do for people. but the idea of trying to keep people healthier longer, and that transition that's happening. And I actually really think that the foundational work that you're doing right now really actually plays very well to that not necessarily just trying to find out how do I treat somebody that's sick, but I think you and I both can appreciate that not everybody that has a bad gene, BRCA1 and 2 is something that was discovered up at the University of Utah. We know a lot about it. Not everybody that has BRCA1 and 2 gets breast cancer.
Yeah.
And so the question is, why do those other people don't? And no one's really been able to no one's really researched that in an extensive way. But your technology and the things that you're building and the foundation that you've built, I think is actually a great transition to start asking some of those questions, as well as is why when I go in and mutate a gene that, why doesn't it universally affect, what are the other things that are involved? And maybe that opens up new possibilities for new ways of developing therapies that don't treat sick people, but therapies that keep people healthier longer. And I see that as getting back to the question of can we cure all these diseases? Honestly, that's probably how we cure them, right? Is by preventing all those diseases from happening and not wait for people. Because I'm of the belief that, you wait for somebody to get Alzheimer's, you're not curing Alzheimer's.
Yeah,
it's too late. Yeah. So we've got to figure out how do we cut these things off before they become a problem. And I think that the approach that you guys have taken and the foundation that you're building right now, I think the adaptation, the next generation of what you're doing is really going to start answering some of those questions of how do we keep people healthy, and how, if you've got this set of genes that predispose you to this, this is what you should be doing now. Because right now we just tell people, it's very dissatisfying as a doctor to walk in and have somebody that says, you have this condition, you have BRCA1 mutation, you have a 60 or 70 or 80% chance of getting breast cancer. What are you going to do, doc? Well, we're just going to watch you and wait for you to get breast cancer. That's not satisfying at all, right? So
no, the medical system in general is pretty frustrating right now. I was in for a sports injury recently, and I answered the same question like 7 times over 4 days, and I almost lost my mind. It was and I'm pretty sure nobody was reading those answers either. There's a lot of work that we need to do across the medical system. I think working to prevent disease is a really important part. I think biology has this, and health has this, we believe that it's really noisy for the reasons you mentioned. Like some people have these mutations, they get sick, some don't. I think the reality is that biology is deterministic. Yeah. If we understood how every gene interacted, we'd be able to say to the person, or how we interact with our environments, we'd be able to say, actually, you have this gene and you probably aren't going to get cancer because you have these other 17 genes or mutations that actually create a lot of benefit for you. Or to someone else, you don't have this mutation, but you probably are going to get cancer. And we have to move to understanding biology as a complete system to understand that level of complexity. We are a ways away from solving that, but we're on this exponential slope. And so my hope, like yours, I think, is that good medicine will be preventing a lot of these things in the future. And the FDA recently came out and said, on this ultra rare genetic disease side with a plausible mechanism, maybe that's a great place to actually take a little bit of risk. Because these are patients who most companies aren't going to work to find treatments for. And so it's good to see. We're seeing progress on many fronts, and we have to stay optimistic because in the background is a pretty broken system.
Yeah. And like I said, I think we are optimistic, and we think that with all the failings, there's still amazing things happening.
So many cool things.
This just the revolution of, I think the GLP-1 drugs have opened people's minds up to say, you know what, this is a hard problem with type 2 diabetes and obesity. We've been hearing about it for years. It is a major, major problem, but we might have a solution, or at least the beginnings of a solution.
Well, those teams, the amount of people who walked away from that field. And then it took some real courage to say, I'm going to stick with this through the research and research and research.
That's exactly.
And now something really exciting comes out. And there's so many stories in medicine and healthcare where it's easy to walk away because it's hard. Yeah. And if you stick with it, eventually pretty good things can happen.
Yeah. No, that's amazing. So I guess let's think futuristically.
Okay.
So where's Recursion in 5 years from now? So the last 13 years or 11 years or however long it's been, you've kind of proved the concept that said hey, we can take this kind of approach. So now that you've proven that and you've built this foundation, what's next? What can we expect?
Well, our hope is that we'll be moving from, these giant robotic automated labs generating data to inform hypotheses to an entirely in silico approach where our wet labs become just the validation of in silico predictions. And this happened in the chip industry. Yep. Back 30 or 40 years ago, computer chips were designed and then tested in a very empirical way. Yep. Now companies like NVIDIA do the entire thing in software, and then they basically just push a button and spend billions of dollars to make those chips in the real world, and they work.
Yep.
Because the in silico kind of physics and electrical modeling is so good. Biology is more complicated, but I think we're going to get there. And in 5 years, I think we'll be at that inflection point. What does that mean for us as a company? Hopefully, many medicines in the clinic. And within 5 years, I hope medicines approved and treating patients, which raises the question of how are we going to commercialize these in this broken system?
Yeah.
And I think that's one of the places where maybe our biggest innovation will come. I think we see the potential for a future where we are generating medicines in an industrialized way that is decreasing in cost over time. If we do our job right, if the machine learns, then it'll get less expensive over time. And if we can do that, maybe there's a way for us to start doing almost like the Amazon Prime of medicine. You're seeing little glimmers of this with Lilly on their GLP-1s, companies going direct to consumers. Maybe there's a membership model for medicines, and we and other companies alone or together could start moving that direction. And, maybe there's no PBMs and those sorts of things. So.
Yeah. Well, I think, I agree with you completely. I think that I've talked to a few administrators of hospital systems and things, and they, the bottom line is we're to the breaking point where things have to change. Yeah. So that's scary and exciting, right? I think it's because it's not clear what's going to be on the other end of this, but I think
something better.
Something better is what I think as well. And the bottom line is we actually can play a role in shaping that as well, because I think the old paradigm of the little company makes the innovation and sells it to the big company. Maybe that's not the way it's going to work in the future. And
I'd be okay with one or two of those.
Yeah, me too.
But just to pay for the rest.
Yeah, sure, absolutely.
But eventually that's where we want to get to.
Yeah, because people probably the most common thing I get asked is why do medicines cost so much? And it's not an easy answer, right? It's such a complicated system that we've developed, and part of it is that it's the way technology develops. We tend to bolt things onto old things, and that's not necessarily the best way to develop new technology. It's usually better just to get rid of the old stuff and build from a fresh foundation. But I think we're seeing that before our eyes. But the good news is that we have a role to play in this, and I think that we can be part of that change, and I think that's what's exciting. So. I think that with all the noise and all the uncertainty and the chaos, and I know you're a CEO of a public company, so you have to pay more attention to things that I do. I there's no stock for me to look up of my companies, but I tend to not try to pay any attention to that because that gets really, really hard. And I guess what is it? The tendency to worry about, because you've got a bunch of employees and they're looking at your stock price every day, and for good reason, right? They're partners with you in this, and they're co-owners in the company. How do you keep people focused on the real mission and the vision to say, hey, the day-to-day stuff, don't worry about that. What do you do as a leader to say, this is don't worry about that. Let's focus on making a difference here.
I mean, I think there's maybe two things. One is we don't talk about it a lot, but we do talk about it, and we talk about it pretty transparently. And so, for example, there were some pretty big swings in all biotech stocks over the last 4 years, but even over the last 4 weeks, it's been more intense.
Yeah.
And so when we were at kind of one of those more existential points, I just got in front of the company and I told them that, we're not going to overreact and we're not going to underreact. And we looked back historically at times when we'd done both and what we learned from it. And I think just having the conversation transparently, all of our employees are really smart.
Yeah.
And being upfront about it's hard, but don't forget what we get to do every day.
Yeah.
Right. And the other thing we do is we make sure that we're in front of patients a lot. Most of our employees have some connection. Like the reason they work so hard on this is that they lost somebody important to them, or they have somebody who's struggling. And so it's close to them in some way. But we make that real by bringing patients in. And I think if you do that in a responsible and respectful way, it is like jet fuel for your team. And it makes a lot of the rest of the stuff feel not unimportant. It's important, people's college funds and all of that. But at the same time, it puts it all in perspective. So I think you just have to be transparent.
I think that makes perfect sense.
And if you tell people not to worry about the day-to-day and worry about the year-to-year, then we have to make progress, and we will.
Yeah,
so
I think that's exactly right. Well, you're obviously, you've learned some things about being a good leader, and I like I said,
so many mistakes along the way.
That's unfortunately how it happens. A lot of gray hair, a lot of scars, but
exactly.
but that's what happens. I try to tell everybody I work with is, listen, the problems are out there with the patients that are suffering and the diseases that we're trying to affect. We don't have to create problems. We don't have to, let's worry about the stuff that is the real problem. And, my mantra is good science is good medicine, and good medicine is good business. So if we can do good science and make good medicines, it's all going to work out. And
a little Henry Tamir there.
Yeah, that's right. Exactly. That's what we try to stay focused on is to say, let's get the science right. Because at the end of the day, it's the beating heart of what we do. And it's really what determines whether we succeed or fail. And I think you've been a pioneer and a revolutionary in an area that and I'm not that old. I'm getting older, but I've seen a couple different waves of things happen, and it's amazing to be your friend and colleague, to see what you've been able to accomplish. And I'm a fan on the sideline cheering for you. And
likewise, thanks for all you've done. You've been leading this space in so many ways.
Well, I appreciate that. We expect and hope for big things for Recursion. Thanks so much for being with us, and always good to see you. I appreciate you being on the podcast.
Good to see you. Thanks, Dave.
That's it for today's episode of Good Medicine. Thanks for joining us as we explore the science and stories behind health, medicine, and innovation. If you enjoyed this conversation, please subscribe to our YouTube channel. That's a great zero-cost way to support us. In addition, please click, follow, and leave a review for the podcast on Apple and Spotify. If you have questions or comments for me, please put them in the comments section on YouTube or reach out on all social media platforms at Good Medicine. Links are in the description. The best way to support the podcast is to check out the sponsors of the show. You can do this by looking at the links in the show notes. That's it for today. I'm Dr. David Beers, and we'll see you next time.